The overall goal of this procedure is to rapidly isolate xenotransplanted human tumor cells from mice to improve the downstream analysis of the desired target cells.
The elimination of the contaminating mouse cells by this method allows the specific assessment of human tumor cell gene expression or in vitro drug responses. The main advantage of this technique is that the human tumor cells can be quickly and easily isolated without having to identify any surface markers on the target cells.
Begin by using forceps and a scalpel to remove the fat and necrotic areas from the tumor sample. Then mince the tumor into 2 to 4 millimeter pieces. Next, transfer the tissue fragments into a single-cell suspension dissociation tube containing the digestion mix, and tightly close the tube. Place the tube upside down onto a benchtop tissue dissociator mount, and confirm that the tissue pieces are in the rotor-stator area. Attach the heater to the dissociator. Then click the folder symbol on the touchscreen and use the up and down arrows to select the appropriate tumor dissociation mode. Next, select the mount position where the sample is located and start the dissociation. When a single-cell suspension has been obtained, quickly centrifuge the tube to collect the cells at the bottom of the tube. Filter the cells through a 70-micron mesh strainer into a 50 milliliter conical tube, and rinse the strainer with 20 milliliters of medium, collecting the wash in the sample tube. Then centrifuge the cells and resuspend the human tumor.